Our next speaker is Morgan Schock. And Morgan was born and raised in Farmville, Virginia, which happens to be the home of the corn cyst nematode, in case you were wondering. She went north from Farmville, almost directly north, to Charlottesville, where she got her undergraduate degree at other university in Virginia. And then she wised up and came to Virginia Tech. And she's working on her PhD in the Barney lab. In the ASCII lab, sorry. In the ASCII lab. So today she's going to give a research proposal seminar.
Thank you, Dr. Eisenbeck. So I'm going to be talking about trying to figure out methods of better establishing gardens of plants that support pollinators. So to start off, if Shakespeare were giving this talk, he might say this quote, which is to say that the original quote is about how what we study doesn't necessarily reflect what all is out there. And so in this sense, what I'm about to show you is that there's a lot more pollinators than you might know off the top of your head. So starting with a point of familiarity, we've got most people, when they think of pollinators, tend to think of honeybees or bumblebees. And then both of those, as well as the blue orchard bee, are available as commercial pollinators where you can actually buy these bees and use them wherever you are. So for honeybees, that would usually be setting up a hive or bringing in hives near an agricultural field. Bumblebees are often used in greenhouses for things like tomatoes. And then blue orchard bees are being looked at for use in apple orchards. And both honeybees and bumblebees are colony bees, whereas blue orchard bees are solitary bees. So you would get a whole bunch of individuals for the blue orchard bee, and you might start with just a queen for the other types. Expanding out, we've also got butterflies and moths are pollinators, and these are all species that are native to Virginia, and you can see there's a fairly large variety here. And then getting even more eclectic, we've got thread-waisted wasps, hoverflies, beetles, and even mosquitoes. These, again, are all species that are native to Virginia that are pollinators. The interesting thing about mosquitoes is that female mosquitoes, as you know, are the ones that will bite you and use blood so that they can provision for their offspring. Male mosquitoes don't drink blood, they instead drink nectar. And that's how they serve as pollinators. So what's the impact of pollinators? Well, 87.5% of flowering plants experience animal pollination. And this study by Allerton et al. Was done, they grouped pre-existing studies looking at pollination of plants by climate into tropical, subtropical, and temperate. And so there's this is the adjusted number for the given skew of the fact that there are more plants and more animal pollinated plants in tropical areas. So there's, it's closer to 90% in tropical areas specifically. So this is adjusted down for accounting for the decreased amount in other places. So this study took a lot of different crop plants and studies that looked at pollination of these plants to see how animal pollination actually improved yield. And so we've got categories down at the bottom, which are explained over here in terms of how much of a difference being pollinated made to yield. And so then the y-axis is number of crops that are in those categories. And so you can see that there's a fairly large number of crops out of this that benefit by at least 10% yield increase for being pollinated by animals. So some examples of that from the essential category are things like chocolate and squash. For the high category, there's apples and stone fruits. In modest, we've got things like mustards and pomegranate. And then things like peanuts, chili peppers, and tomatoes don't actually benefit from pollination very much. And then things like olives and grapes, it doesn't seem to make much of a difference at all. So in this study, they were looking at seed set in apples, which is a measure of productivity and yield. And so the diversity of bee species present correlates with yield increases. And wild bees are more important than honeybees. So what this is showing is we've got bee species, wild bee abundance, and functional yields is just groupings of bee species. And then we've got honeybee abundance. So you can see for these three, there is a positive correlation between these aspects and seed set. But in honeybees, there is not actually a correlation. And some of that is because in a lot of apple orchards, it requires cross-pollination between one variety of apple to another. And so they'll have 2 to 3 rows of the apple variety they're growing, and then a row of the one that needs to pollinate that. Honeybees tend to go along rows, and so that doesn't actually help with getting the pollen where it needs to be, whereas wild bees will go all over the place, and so they're more effective at actually effectively pollinating the apple trees. So there are different ways that we can intervene between domesticated and native pollinators. For domesticated pollinators, in particular honeybees, we have their hives in a way that we can access them, we can monitor them for fungal infections, we can monitor them for mites, and we can manage their honey levels so that they don't attract things like mice and badgers. And we can even breed them for resistance. But for native pollinators, their nests tend to be inside plants or in the ground, and so they aren't accessible. And there's a lot of solitary species. So for a hive, the loss of even 100 workers of honeybees isn't that big a problem for a hive because the queen can always lay more eggs. And the unit of eusocial bees is a hive more so than an individual bee. Whereas for solitary bees, if a solitary bee dies, that bee does not procreate, and therefore the population as a whole is affected a lot more by each individual bee dying. And so also for butterflies, they're like the monarch butterfly migrates over huge distances, and so you can't foster that population in just one location. You have to cover the entire area that it goes over. And then there's just such a variety of native pollinators that it's hard to address all of them at once. But the thing that we can do is manage food resources for them. We can plant things that have pollen and nectar, which is what they need. So in trying to create a list of species to work with, I cross-referenced between the USDA plants database and the Ladybird Johnson Wildflower Center to try and find plants that fit criteria of being native to broad ranges of Virginia. So this is the distribution of one of the species that I worked with, Lanceleaf coreopsis. And you can see green counties are where it is naturally found in Virginia, and white counties are where there isn't data of it being there. It doesn't necessarily mean that it's not there, it just means that no one has reported it. And then the Ladybird Johnson Wildflower Center had the information on which pollinator species these plant species actually served. And so comparing bloom times and how likely it is to attract a deer, so these plantings are being planned for rest areas and roadsides. It's very important that they do not attract deer because that would increase accidents. We want to create a list that has coverage over an entire season of activity for pollinators. from the very early spring through to the late fall. And so also having this diversity, well, in addition to being necessary for having continuous food resources, because these plants no individual species tends to have flowers through the entirety of that time period. Having a more varied species mix makes the garden more resistant to invasive species, and having a greater diversity of plants supports a greater diversity of pollinators, because different pollinators look for different things in flowers. So there's long-tongued bees and short-tongued bees, and hummingbirds need something different from either of them. And so to address a wide variety of pollinators, you need a wide variety of plants. So the problem with establishing these gardens is that it's very hard to get them started from seed, because when you plant these plants, these weed seeds that are already in the seed bank, they will outcompete the plants that we're trying to grow, which are fairly slow growers at the beginning. And so without management, it's very hard to see change happen, and a lot of effort to make these gardens. And the other alternative is trying to start from whole plant transplants, but that is very labor intensive and expensive. So we want to try and improve this from seed establishment. And the way that we're looking at doing that is with activated charcoal siphoning. So this has previously been used in establishing native grasses that have the same problem of having a very slow start to their establishment. And the way that this works is so similarly to this English muffin. Activated charcoal has lots of nooks and crannies in it. And so you can keep it in air, water, or soil, and it will trap things that would be considered contaminants. In this case, it traps the herbicide that you spray over the top. So the way that we would be applying it is with a banded application. So we have a row of seeds that we planted, and right over top of what we planted, we have a band of charcoal. And then over the top of that, we can spray a pre-emergent herbicide, which prevents weed seeds in between those rows from germinating, so that the seeds that we want to have grow have a bit more of a head start and advantage and are able to establish better. So I'm going to be going over 3 trials that I've completed the greenhouse preliminary trial, and then I'm in the process of doing the 2 field trials. So for the greenhouse trial, it was a proof of concept that the activated charcoal did siphon the plants effectively. And so this is a GIF of how Dr. Askew was applying the charcoal. So activated charcoal, we had to suspend it in water in order to spray it like this. And on its own, it will not suspend in water for extended periods of time. So it was our first attempts were requiring continuous agitation of the spray liquid. But what I ended up figuring out is that by using a small amount of xanthan gum, which is used as a thickener often in gluten-free baking, it keeps the charcoal suspended within the liquid so that it can be sprayed with an even dose, and so we know how much we're actually putting down. So these 2 passes was enough to put down 300 pounds per acre of charcoal in this band. So we put native soil into these pots, and we had 4 activated charcoal treatments. We had no activated charcoal, 300 pounds per acre over the top, 600 pounds of acre over the top, and then the last one was a split application where we put down a 300 pound per acre pass of charcoal, and then we put in the seeds, and then we put in another 300 pounds per acre over the top of them. And so the species that we were working with for this were Lancet coreopsis, bee balm, bearded beggar ticks, partridge pea, and New England aster. And so these are all species that are pollinator serving and native to Virginia, and the preliminary results from that, we saw better germination from the larger seeds, but which would be the partridge pea and Lancet coreopsis especially. But we think that part of the problem with that was the way the soil was pumping, and so the small seeds were falling down into cracks and thus not getting exposed to light like they needed to germinate. And the highest rate of activated charcoal did prove to be the most effective, and the split rate seems to be more effective than the only over-the-top rate of 600 pounds per acre. And we also saw that some of the species involved do exhibit tolerance to the herbicides we were using because they were growing even in the pot with no charcoal protection. For the first field trial, this is a representation of a single rep within the trial, and there were 4 reps at each site, and there were 2 sites. So there's 2 charcoal treatments for each herbicide sprayed. And so we have the 600 pound over the top is represented with the solid, and the split application of 300 in furrow and 300 over the top is represented with the bands. And so we used 8 different herbicide treatments, including an untreated check. And we used a slightly different composition of plant species because we wanted to maximize the number of plant families that we were covering with this experiment. And so we kept the Lancet coreopsis, the bee balm, and the partridge pea, but we also added in foxtail beard tongue, and golden alexanders. Unfortunately, I didn't see either of those germinate in either site for any of the treatments. They might in the coming year, because that's a thing that does seem to happen a lot in these plantings, is that you don't see the plants the first year, but they show up in the second year. And so we're going to continue monitoring these sites. But and the herbicide applications that we did were, these are all different pre-emergent herbicide treatments. And the most effective that we saw so far is the plateau. It does a very good job at leaving the plants that we want to have there alive while clearing out weeds from the other spaces. So here we've got the in-furrow and the over-the-top charcoal applications of the untreated plots. And here we have the plateau treated in-furrow and over-the-top for one of the replicates at one of the sites. The 2 sites for this one were the rest area at Ironto, and the other was the rest area up at Rocky Gap, almost on the West Virginia border. And so here's a picture of the Ironto site. This is in June, and then this is into August, and then this is fairly close to now. So you can see that we did have quite a lot of the partridge pea came up, and the Lancet coreopsis also was there. It's these plants here. The other field trial site was at both of the Radford rest areas, and for this one we're replicating over time and space, so each trial site is a single rep, and we're planning to, so we have 2 reps so far. So it's 16 different herbicide treatments, including an untreated check, and then there's 20 different plant species. So there's 20 rows going across horizontally in each of these plots. And within each plot, the order of the plant species is randomized. And so here's the plant species that we have in this, or the way I came up with this list was cross-indexing through a lot of different pre-existing pollinator supporting mixes of what species they included, and trying to select ones that were the most prevalent across all of these different mixes. And so here's what those look like. And so we've got a fairly wide variety of families here. We've got some legumes that would also help the overall mix with their nitrogen fixation, and we've got some mint family, and most of these are asters, because that is just a very large family with a lot of representation in these mixes already. And so the herbicide treatments we have here, in the other experiments we have these combinations, but we don't have the pendulum aquacap, gallery tower, and sureguard on their own, and so we do in this study so that we can tease apart what is effective of those mixes, and see if just see what's going on. So going forward, we are going to establish 2 more trial sites of the 2 that are at Radford, so that we will have 4 reps overall. We're continuing to look at how things are doing in these sites, and using this result, we're going to narrow in on a specific combination of herbicide, charcoal treatment, and a broad mix of species that will be most effective at easy establishment of a wide variety of plants. I'd like to thank my advisors and lab members and everybody else who's helped and been involved. And is there any questions?
Any questions?
Why not just use an herbicide like a Mazepec that is this really, I work in wildflowers too, we use that in our plots, so why wouldn't you just use that in this situation?
I do believe that plateau is a Mazepec.
Okay.
I'm pretty sure it is. And so yeah, and we do use it, and the reason that we included it is because it is so prevalent in wildflowers already, and the fact that there are already wildflower mixes that are tailored towards wildflowers that are already tolerant to a Mazepec. And so that also was something of a consideration in trying to pick species, was that it is just another advantage that we could exploit in trying to get the best possible results. And so, yeah, that we did use a Mazepec.
So, how affordable is activated charcoal? I know in like small quantities it can be pretty expensive. Is there like a reasonable source of it?
Yeah, there are bulk sources. I bought an entire pallet load of at least 600 pounds for $600. So that's a very cheap rate compared to trying to buy it in smaller amounts. And it, like I said, at least 600 pounds. It could be up to 1,000. We didn't actually weigh it. It's a giant bag of activated charcoal in a square.
Any more questions? All right, let's thank our speakers.